Hey everyone, Dr. Tom Moorcroft here. Thanks for joining me. Really appreciate it. Today I wanted to talk about Lyme disease and the possibility of persistence in Lyme. As you know, there's a lot of discussion all over, in medical circles, on the internet, even in other videos I've done talking about, is Lyme persistent? Is a persistent infection the cause of your symptoms, or is it really just some sort of post-Lyme inflammatory process? So, there's a lot of research going into this now, and that makes me really hopeful that we're going to continue to get some really good answers out of this. But from what we know now, there's a lot of research that's been going on that really does support the probability that Lyme can cause a persistent infection. A lot of this research is being done by Monica Embers and her group out of Tulane. There's also some work being done by Dr. Zhang over at Johns Hopkins. And so it's been kind of interesting to read through these things. Dr. Embers has a paper out about the persistence of Borrelia burgdorferi in rhesus macaque monkeys following antibiotic treatment of disseminated infection. And basically what they talk about is they took some monkeys and they infected them for 4 months. And then they took a group of them and they treated them with doxycycline for a month. And they did this treatment, and the blood test went from positive to negative, and then they sacrificed the animal and did tissue studies as well as some other advanced diagnostic studies, and found that actually all of these monkeys still had viable Lyme disease spirochetes in them. So 28 days of treatment for Lyme in these experimental animals cured 0% of the monkeys. Now they did another study where they did 4 months of infection, they did a month of IV antibiotic with ceftriaxone, and then 2 months of doxycycline. And in this study, the blood test became negative, and then when they sacrificed the animals and they did tissue studies, they found that 27% of those monkeys were cured of Lyme disease to the best of their ability to find out. The problem, though, that means that 73% still had viable infectious Lyme disease in their body that could be causing symptoms in them. And another interesting sort of piece of that study was that when you look at the control group, the non-treatment monkeys, half of the non-treated animals, their blood tests went from positive to negative over the course of the study. So even without treatment, this previously positive antibody went to negative. So in one of the other studies they did at Tulane, they looked at, they artificially infected monkeys again, and they treated them with 28 days of doxycycline, and they also had a control group. And things that they found were that, the host-dependent signs of infection, things like rashes and other symptoms, as well as their antibody response were variable based on the unique individual that was infected. And so some of these monkeys had blood test positive to certain proteins in Lyme, but not to other proteins in Lyme. Some had tissue samples, like skin biopsies, positive. Some had them negative. Some, when they put a sterile tick on them to feed, some monkeys infected the tick, some monkeys did not. So there's a lot of this going on where there's a variability based on, the individual that was infected. The other thing they found is that after this 28 days of treatment with doxycycline, they had evidence of persistent, intact, viable, metabolically active Borrelia burgdorferi, Lyme disease bacteria after this treatment. So, that's problematic that all of these, monkeys that were infected remained infected after this month. So that kind of confirms what they found in their previous study. And then they said also that the persistence of specific antibody production may not really reflect the actual infection of these animals, because again, when they do these tissue samples and when they do these advanced diagnostics, they're finding that these animals are still infected. So that's problematic. And it's really interesting when you look at the charts from this paper. I mean, you just see that some of the organisms are positive at 7 months, which is, so they got infected for 4 months, then they got treated for a month, and then at 7 months, they're positive. And other animals are negative at 7 months, but they're positive at 12 months. And so there's a variety, and this changes depending upon each monkey, at what point you do the testing, and what part of Lyme you're looking for. So there's just this variety is all over the place. We're often asked the question, well, why does this actually happen? Well, One of the things are bacteria such as Lyme can hide inside of cells. And so we call that intracellular localization. And there's a lot of evidence that this happens. So, if your immune system's going after these organisms and they go and hide inside of the cells, sometimes even the cells of the organism that are looking for them, they're not going to be found. And also, then you're not going to have an antibody response. The other thing is they can be sequestered in these antibiotic privileged sites. So basically they go to areas that antibiotics and other things like antimicrobial herbs may not be able to get to. So that's one of the reasons that Lyme might persist. The other thing is there are forms called, dormant forms, kind of like one is called a cyst. It's almost like the spirochete rolls up into a ball and insides out, and then it's metabolically inactive and it's kind of protected itself, almost like an armadillo rolling itself up. One of the other forms of protection we know of is called biofilm, where we have a bunch of these organisms get together and they sequester a lot of fat and fibrin and polysaccharides and protect themselves, from antibiotics. And some of these biofilms can be quite strong and prevent the antibiotics from getting through and killing the organism. So it's a protective measure. So these things are two of the main players in sort of the direct persistence of this organism. Then the other problem is the more and more we research Lyme and associated diseases, we find that both ticks and those who are bitten and infected by ticks have co-infections, or multiple infections. So, maybe a deer tick is carrying Lyme disease and Borrelia miyamotoi and Anaplasma and babesiosis, maybe even Bartonella henselae. And then this gets transmitted 2, 3, 4, or all of these organisms gets transmitted to the person who's bit. So now the problem is when we develop tests, we actually develop tests assuming that there's only one infection. And so, we don't have tests for Lyme and Babesia. We have tests for Lyme or Babesia. And no one's really studied what happens if the organism infected has 1, 2, 3 different infections at the same time. And so maybe some of the reasons our tests aren't so good is because we have too many infections going on for your immune system to respond properly. But one of the reasons the infections may persist is because now there's a multitude of infections, many of which are going intracellular, many of which are suppressing your immune system, blocking detoxification pathways. And so work out of Yale has suggested that, Lyme and Babesia co-infection in the environment, in the host, like the mice, as well as in the ticks, is much more common than expected. And then also that when people get infected by more than one tick-borne infection at the same time, it can make your symptoms more severe. It can make it more difficult to diagnose because now you have symptoms looking like many different diseases rather than one. So you kind of get that combo platter, so it's not as crystal clear when you go see the doctor. And then the problem is the treatment is also harder. So I'm going to throw some of the links to those articles below so that you can see what I'm talking about, because I think it's important to understand that when we talk about these things, this is all based on science. And I think it's a lot of times what I'm hearing, I'm talking to people, they're like, that, Lyme doesn't persist. And I'm like, well, how do you know that? And they're like, well, it doesn't. That's what I've been told. And I'm like, well, have you looked at the research? And, while the research isn't definitive at this point, there's mounting evidence that persistence is very probable. And certainly in experimental animals over and over, we're seeing it. And thankfully we're not, I can't say how much I appreciate these animals, and the studies that are being done on them because, we can't do it on people. I wish there was a better way to not even do it on animals, to be honest with you, but we're getting so much information that explains why so many people are suffering and will help guide us to that next place of how can we really, eradicate these infections. And that work's being done. Dr. Zhang at Johns Hopkins is looking at antibiotic combinations and in figuring out how we can actually maybe hopefully in the near future eradicate these infections. But in the meantime, we have to understand that the best evidence we have is really looking at these things being Lyme in particular
having a high potential for persistence in the body and creating chronic infections. So please take a look at some of the links below, because it'll get you over to the articles that support the things that I've been talking about. If you have any questions, please let me know in the comments below. Let me know if there's things you want me to address in the future. I really appreciate you joining me today. Thanks so much.